Good day, everyone, and welcome to Introductory Microbial Ecology class today. Today, I'll be talking to you about microorganisms and the ecological theory. Microorganisms and the ecological theory. The study of microorganisms in the natural environment is called microbial ecology. Microorganisms are important parts of the ecosystem, and they play major role in energy transformation and biogeochemical processes. Specifically, microorganisms are involved in synthesis and degradatory processes. Some of these microbes are autotrophic. That means they are capable of generating new organic matter from carbon dioxide, while others are organotrophic and therefore participate in the biodegradation and recycling of organic and inorganic matter. Microbial ecology primarily deals with two aspects of microbiology of an ecosystem, which are the following. Number one, the types of microorganisms present in the ecosystem or the environment. Number two, the nature and the extent of their metabolic activities. Number one is the types of microbes, that means who is there. Number two is the nature and extent of their metabolic activities, that means what are they doing there. The microbes that are there, what are they doing? So there are two things microbial ecology is interested in. Who is there, that means which microorganisms are in the environment. Number two, what is the microorganism doing in the environment? The presence of an organism in an environment is not of overriding importance, but a knowledge of their activities. Essentially, only an active microorganism have an ecological impact. Specific biochemical transformations serve as good evidence of the presence of specific bacteria and their metabolic activities. What this is saying is that the microorganisms present in the environment are important. It's important we know them. But if the microorganism in the environment is not doing much, it's not beneficial, it's not harmful, it's just in the environment, we do not need to pay much attention to that organism. But a microorganism that is doing something very good to the environment, we need to pay close attention to that organism. And that is the direction that most research in advanced countries and even in developing countries are going today. Take, for instance, the issue of the coronavirus. It is in the environment. It is a virus which is infectious. There are other coronaviruses, but the COVID-19 is a strain that is different from others and is causing death. Now, because of this, you can see the way the whole world has gone and everywhere has changed because of the importance of COVID-19. There are other microorganisms like Escherichia coli, Bacillus subtilis. There are so many other organisms. Even the HIV virus is also there. But the organism of attention at the moment in the whole world, as far as 2020-2021 is concerned, is this virus, the COVID-19. Natural habitat of microorganisms are diverse, but any habitat suitable for the growth of higher organisms will also support the growth of microorganisms. In addition, microorganisms are found to flourish in extreme conditions where higher organisms cannot. Microorganisms are known to have established various forms of relationships with plants and animals. The number and composition of microorganisms in a habitat are governed by some factors, and some of the factors are temperature. The temperature of the environment will determine the type of microorganism you find in the environment. The pH of the environment will determine the type of microorganism you find in the environment. The water availability of the environment will determine the type of microorganism you find in the environment and in what proportion. The oxygen concentration of an environment will determine the population and the type of microorganism you find in an environment. Also, the nature and concentration of nutrients will determine the type of and the composition of microorganisms in the environment. So microorganisms do some things in the environment. Some are producers, some are consumers, and some are decomposers. Now, isolation and identification of microorganisms go hand in hand with activity measurement in order to generate a more complete picture of the microbial ecology of a given habitat. An environment usually contains a variety of microbes. For this reason, methods and procedures have been devised to permit the isolation and culture, that means the growth of microbes of interest. What we're talking about now is methods in microbial ecology. The most common approach is the enrichment culture technique, in which a medium is a growth supporting material. A medium is a growth supporting material for microorganisms, in which a medium with a set of incubation conditions are used that are selective for the desired organism, which are counter-selective or inhibitory for the undesired organism. In enrichment culture technique, you want to isolate organism A, but in the environment, organism A, B, C, D, E, F are in the environment. If you use a general purpose medium, what you are going to isolate is going to be the organism A, B, C, D, E. But you are interested in organism A. So you find out the medium that only organism A can thrive in and other organisms cannot thrive in. So it is that medium you use in the isolation. The moment you use that medium in isolation, it's going to eliminate organism B, C, D, E and only allow the growth of organism A. The most common approach is the enrichment culture technique, in which a medium, that is a growth supporting material and a set of incubation conditions. Apart from the medium, also incubation condition. If incubation condition is for most organisms is 37 degrees C in terms of temperature. Some organisms, let's say organism A, can grow at 37 but can also grow at 44. So you can raise the temperature of the medium to 44 degrees C, where other organisms B, C, D, E cannot grow. So it will allow only organism A to grow. It may also be pH. Organism A can grow in acidic pH, but other organisms, they need neutral pH. By the time you make the medium to be acidic, what will happen is that only organism A is going to thrive. Talking about other things like oxygen or the oxygen level. If organism A is anaerobic and the other organisms in the environment are aerobic, you make the growth medium to be anaerobic so that the aerobic organism will not be able to grow. Only the anaerobic organism, in which organism A is, will be able to grow, and then you can easily identify the organism, which are selective for desired organism, which are counter-selective or inhibitory for the undesired organism. The appropriate inoculum must be used. Inoculum is a sample that you need. For example, if you are suspecting that somebody has diarrhea, you take sample of feces for diarrhea to be able to see which organism is causing. Is it Vibrio? Is it Entamoeba histolytica? Or which other organism is responsible? That will help you to do the microscopy, to do the culture, and then the sensitivity to know the antibiotics that the doctor is going to administer to the patient. It is generally believed that everything is everywhere, but the environment selects. Enrichment culture are often obtained by placing the sample or culture component into a highly selective growth medium. Another technique is the use of the Winogradsky column. This column is designed for the isolation of purple photosynthetic bacteria and other anaerobes. It was originally designed for the study of soil microbes. It is a miniature anaerobic ecosystem. The Winogradsky column was originally designed for the study of soil microbes, or for the study of microbes in soil. So, Winogradsky column was designed by Sergei Winogradsky. It's a column, vertical column, cylindrical column, which soil is put in. At the base, it is sealed, so there's no how air can reach the base. Air can reach the top because you cover it with polythene nylon, white polythene nylon or transparent polythene nylon. Now, whether you make 2 or 3 or more holes, or you leave it open, what you are going to obtain is that an amount, the upper part is going to contain more air or more oxygen than the lower part at the bottom. The upper part to contain more oxygen than at the bottom. So even whether the gastric column is left open or not, the anaerobic organism will run down and go and settle down, especially if the column is longer, is long. Before air can transfer to the bottom, it's going to take a lot of time and may even be difficult. So the anaerobic organisms will go down and settle at the bottom. Now, in nature, what can we term as example of winographic column in nature? Now, the soil on which we stand. The soil on which we stand is an example of a winographic column. When you stand on the soil surface, the upper part is like the upper part of the column, where we stand, where we move, where we walk on the soil. Now, on that part of the soil, if you take a scoop of that soil and you go and look at the microorganisms at that level, you will find most of them are aerobic. Why? Because that is where air can reach. As you go down to the soil, 1 meter, 2 meter, 3 meter, 4 meter, 5 meter, this type of soil component, the color of soil is going to change, just as the color of soil is going to change in the winographic column. The color of soil, as you go deeper at the layers of the soil, you will see that the soil color are going to change in nature, just as they change in the winographic column. If you are looking for anaerobic organisms, sulfur oxidizers, if you go down deep into the soil, you will find them. You cannot find plenty of sulfur oxidizers on the surface of the soil in the natural environment. The reason is because air is available. So once air is available, putrefaction cannot occur. And when putrefaction does not occur, sulfur oxidizers will not be able to predominate. So you can check my other materials. I talk about the winographic column. Check other materials that I have that highlight talk about the winographic column. Now, let me just give some of the things that happen inside of the winographic column. When the winographic column have been prepared, the cylinder is left to develop for weeks, while placed where it can receive adequate sunlight. The expected observations in the winographic column are as follows. Typically, algae and cyanobacteria will quickly appear in the upper portion. They help to keep that zone aerobic by production of oxygen because they are photosynthetic. Purple and green patches appear on the outer layer of mud exposed to light. The purple portion consists of purple sulfur bacteria, while the green has green sulfur bacteria. Both populations develop as a result of fermentative decomposition processes, which lead to the production of alcohols, organic acids, and hydrogen, which are suitable substrates for sulfate-reducing bacteria. One advantage of the winographic column is that it is a ready inoculum for enrichment cultures, either aerobes or anaerobes. It can also be used for biodegradability studies, in which case the substance to be studied is spiked on the column and the organisms which can degrade it is then selected. With this column, it is also easier to observe the different physiological groups among organisms in the column than in an enrichment liquid culture medium. Now, we have said that this winographic column can be used to get inoculum for enrichment cultures, either aerobes or anaerobes. The question I want you to answer on your own is that in the winographic column, which section are you going to find the aerobes and which section are you going to find the anaerobes? Is it the upper part, the middle layer, or at the bottom of the column? Example of monographic column in nature again is a pond. Is a pond. Is a pond. At the bottom of the pond, a lot is going. At the surface of a pond is stagnant water. And in preparing the monographic column, it is static. It is not moving. Monographic column cannot, an example of it is not a flowing river. A flowing river will not give you an example of monographic column. In a flowing river, where you can find a monographic column is a section of the river where there is not much movement, where the river is not flowing. And we may have that in some rivers, in some sections where the water just branches off and is not flowing. In such sections, you may have the column. In a flowing river, the microbes are always changing. They are always changing, even though we have idea of which microbes can be at which place at which time. But in a pond, the water is static, so it can form the layers. Everybody takes its place in a pond. But in a river, the organism in a river mostly are aerobic, especially at the surface. Mostly are aerobic, and even the one at a river is wide and is flowing. Some of the water that are down, organism down, they come up. Some of the ones up, they go down. They keep mixing. So it is quite difficult to compare a monographic column with a flowing river. Remember, in a monographic column, it is not flowing. So a good example is in a pond. A pond contains mud. It can even be in a marsh. In a marsh environment, the surface will be quite different from the other parts as you go down in a marsh or in a pond. Now, let's talk about the chemostat enrichment culture. Enrichment cultures can be carried out in a chemostat. That is, the procedure of growing and isolating microorganisms in a laboratory can be carried out in a chemostat. A chemostat, what is a chemostat? A chemostat is a continuous culture device that allows for the manipulation of both the population, density, and growth rate of the culture by controlling certain culture parameters. For example, temperature, pH, nutrients, etc. For use in enrichment culture, the chemostat is inoculated with a suitable inoculum source and run at a defined dilution rate. Eventually, the organism that is best adapted to the conditions prevailing is favored. A major advantage of the chemostat as an enrichment device is that it allows the study of microbial competition at a low substrate concentration, similar to what we encounter in nature or in a pond. Competition for nutrients in the chemostat will enable us select from the original mixed population the organism that is capable of growing fastest as the limiting substrate concentration and the dilution rate chosen. Other organisms are eliminated because they cannot compete. What you do in a chemostat is that you give a particular amount of a particular condition. I've mentioned that before. Let's say temperature. You may use temperature as a condition. You put temperature at 37 degrees C. Some bacteria grew. At 38, the number reduced. At 39, the number reduced. At 40, the number reduced. At 41 degrees C, the number reduced. At 42, the number reduced. You see, as you keep changing the temperature, you are reducing the microbial load in the culture, and by so doing, you are able to know the organism that can be able to thrive at a particular temperature. You can also do that with pH. You can even do that with nutrients. You can do that with nutrients. You can, if you are using, for example, mineral salt medium, instead of using potassium, you may leave out potassium and see when I put potassium, how many organisms did I have? Which organism were present? When potassium was taken out, which organism also was present? Also, you can also use sodium chloride added to mineral salt medium. Sometimes you may decide, okay, let me see if I increase the amount of sodium chloride in the mineral salt medium, what is going to happen? Which organism are going to run away? Which organism will not be able to grow? These are some of the culture parameters we're talking about, and there are many more. In ecological studies, chemostats have served as useful models for microbial growth in nature under conditions of low nutrient concentration, and they have found special use in studying bacterial growth in lakes, rivers, and seas where concentration of organic substances are usually low. So take note, in lakes, rivers, and seas, concentration of organic substances are usually low. So I want you to find out the reason why concentration of organic substances are usually low in lakes, in rivers, and in seas. The objective of an enrichment culture study is to obtain a pure culture. A pure culture is a laboratory medium maintaining only one type of microorganism. Now, if you have different type of microorganism isolated from the soil, you can have a pure culture by only having one type of microorganism on a plate, on a petri dish, on a medium in a plate. When you have only one type of microorganism, that plate, that culture medium is called a pure culture. So I take that again. The objective of an enrichment culture study is to obtain a pure culture, that is a laboratory medium containing only one type of microorganism. A number of ways exist in microbiological techniques for obtaining pure cultures. Now, I want to look at measurement of microbial activity in nature. Three methods are common in microbial ecology for measuring microbial activity. These include the use of radioisotopes. Another is the use of microelectrodes, and we also have the use of stable isotopes. There are so many others that have been developed today, so I want you to check on that, and let's have them in our WhatsApp group. Check on that and mention them in the WhatsApp group. The first, which is the use of radioisotopes, is extremely sensitive, so that it can measure the activity of metabolically active organisms that are present in very small numbers in an environment. The activity of microorganisms in natural environment can be assessed by measuring the uptake of the radioactively labeled substrate. So, in the use of radioisotopes, radioisotopic tracers for measuring specific chemical processes are widely used. For example, if the process of photosynthesis is to be measured, the uptake of carbon dioxide, which is labeled, instead of using carbon 12, you can use carbon 14. So you use carbon 14, that is labeled carbon dioxide. The uptake of labeled carbon dioxide into microbial cells can be measured. Similarly, if surface reduction is our interest, the rate of conversion of sulfate to hydrogen sulfide can be assessed. In many natural environments, methanogenesis is common phenomenon, and these can be studied by measuring the rate of conversion of an isotopically labeled carbon dioxide into methane. So you label the carbon dioxide of interest. The carbon dioxide that you're interested in, you label it as carbon 14. But you know the normal carbon dioxide contains carbon 12. So when you label that, by the time the reaction is going on, you will now be looking for carbon 14 which have been labeled. So you can easily find it and see it and be able to monitor what is happening. But if you use the normal carbon dioxide, there's no how you can really be able to see what you are supposed to see in that. So it depends of a suitable reductant like hydrogen. So we are looking at CO2 plus 2H2, will give us CH4 plus O2. So we're looking out for the methane, the CH4 that is going to be produced. Now, what is autoradiography? Autoradiography is used to study the activity of single microbial cells. Specifically, it is used to measure the uptake of any substrate available in radioactive form, including organic amino acids. Now, what are microelectrodes then? Microelectrode consists of small glass electrodes used to study the activity of microbes in their microenvironment. The common ones are the ones used to measure the pH of water, pH of oxygen, and sulfate. The electrodes, when being used, are carefully inserted into the habitat using a micromanipulator that allows for the precise movement of 1 millimeter or less. The use of stable isotopes is noted for the study of biogeochemical transformations, which are often carried out by geochemists in alliance with microbiologists. Particularly useful is this method for the study of carbon and sulfur in natural environment. So with this, we are going to stop on this note on microorganisms and the ecological theory. The question was asked some students, where can you find Winograsky column in nature? Where you can find Winograsky column in nature is you can find it in a pond, or even in the normal soil. In the normal soil, we know is a good example of Winograsky column. But in a normal soil, you are standing on the upper part. Which you cover with a foil, or which you cover with a polythene nylon, which is transparent. So if you want to see what's happening in nature, you have to dig down and dig down. So you dig down, the color of the soil, you see them keep changing. And if you take the microbial, the soil sample and check for the microorganism there, and test for sulfur and test for other things, you'll find as you go down, the sulfur content keep increasing because of the microbial activity that are going there. So this is where we are going to stop for this lecture at this time. I believe that you are going to study very well and read other materials, both online and offline. You can check your Prescott Microbiology have some materials. There is this material by Paul Singleton, which he wrote about his bacteria in medicine, biotechnology, and biology. Thereabout. Read on that and read other materials too. By the time you do this, you're going to find more material. You can check out for Nesta and Nesta. You can check out for Talaro. You can check Brock's Microbiology. These are textbooks that are going to help you to be able to know more about microorganisms in nature. So have a good time and enjoy microbial ecology. Bye-bye.